I'm so excited we'll get to hear straight from Xiangting all the amazing scientific work we've done. So just join me in welcoming Xiangting.
Thank you all for being here and joining through the Zoom. I hope through the Zoom you could hear me clearly and also see my presentation slides. Thank you. Okay, so my name is Xiangting, and today is my defense for my thesis, which is light-induced assembly and repeatable actuation in calcium-driven chemomechanical protein network. Let me start with the basic structure of the cytoskeleton. It is the same as the skeleton to support our body. The cytoskeleton is a building block to support the cell shape and help them to move. Here is an example. If you can see my notes here, looking at this amazing picture here, what happens here is you can see the cell is about to divide. And how does it do that? It using the web of the filaments, the actin, which is color-coded right color here, and on the web of this bright color active filaments actin, there is the motor. It's called myosin, and it's coded in the blue color here. And what happens here is the motor is moving on the actin filaments and helps the cell to divide into two. Another example I like to give is the right picture I'm showing from here. You're seeing here is like a really long filament. It's called microtubulus. It's another major component for the cytoskeleton. It is much longer and rigid. And what you're seeing here is a motor. It's called kinesin. It is a motor carrying a much bigger than size vesicle to just to work on the microtubulus. I always like to give an analogy you can think about. It's the highway and all the motors is a vehicle and driving and helping the transport here. So the cytoskeleton, it is where the traffic is happening inside of the cell. I'd like to have a little bit history of this scientific journey for the actin-myosins. It is around 80 years from 1942, and it's still going on. So first, they kind of extracting the actin from all the cells, and now what they do is like they have the single cell, and they can do the single cell assay testing for its motility. And recent several, like a decade, people start to reconstitute it and trying to use a bottom-up way to study the actin, myosin, or the microtubule systems. I want to give an example here. It is, I want to elaborate a little bit more of this bottom-up system. So for the bottom-up system, what people want to, what I mean is like people are saying, we could only understand what we can build. So what they want is like they want to know what is the essential component or the minimum component to make a cell function and to keep the cell just function normally. And I like to give an example. Here is an example. What they do is, if you can see my mouse, here what they do is like they put a light switch in to the motor system, and so that in this way they could use the light to control the polymerization of the actin-myosin, which I'm showing here in the middle. And another example for the light-controlled system I'm showing here is called microtubulus. So you can see the microtubule with the yellow light controlled in the center, you can see is polymerizing and forming these beautiful esters here. And in this way, we could understand all this collective behavior and property in a much more controlled way. Over a decade, our group have worked on a non-actin-myosin based molecular motor system. It is from a cell. It's called a spiral osteoma. So you can see what happens here is the spiral osteoma contract really fast in milliseconds. What I mean millisecond, I always use this analogy. So it means just within the blink of eyes, you did. It already contract 20 times. It is really fast and fascinating. And what support this mechanism is the using a preformed mesh network is called myelin. And I always say it's an active layer between the membrane and the inside of the cytoskeleton. You can say it's kind of like a fishnet network here. And they can help the spiral ostrom to twist 70 of their body length in millisecond so that it can generate enough momentum for them to hunt for the food or just chase for the prey. More exciting, we find out actually the myelins are much powerful compared to the actin or the motor system I have described in the earlier stage slides. It is even more powerful than the combustion engine we made. So what we are interested in is okay, so if we zoom in to see what happens, more fascinating, we find out the whole contracting process or the whole contracting motion, it is driven or triggered by the calcium and no ATP required in this contracting motion. Let me help you to just go through these beautiful schematics one by one. If you can see my mouse here, what happens is the calcium here, we model the myelin as a spring. And what happens here is like the calcium wave travel along the whole body of the cell. And the stretching of the spring, it will contracting to the local contraction, and then it will make the whole body, whole cell contract. And I like to emphasize one more time, no ATP is required in this contracting process. It's only triggered or only powered by the calcium.
I think you have to admit someone and then you will start work.
Oh, okay. Okay, so in this process, is no ATP is required and it's only calcium driven and powered. And actually, it is really interesting to me because for me, it's like I always thinking about calcium urolate. It is just a primary signaling agent in the cell. It's like during the physiological calcium signaling process, it is start from like a really low calcium concentration inside of cell. It's around like 100 nanomolar, and then it will increase directly to 1,000 nanomolar, just depending on different stimuli and factors, for example, the autophage or like the proliferation and all the same. One of the quite famous pathway I like to mention here is like the muscle contraction pathway is like it's called the myosin light chain pathway. Using this example, maybe I could help you guys to understand a little bit more of what the calcium doing here. So calcium, it is more like a signaling. I put it, it's a traffic light here. And it will give the signal to the chemomechanical, and then the chemomechanical will just triggering and using the ATP to transmit the signal to the neck muscle. And then they will just start to the whole muscle is like it's kind of click behavior to use to make the muscle contraction. And in this whole process, they're using the ATP to trigger and everything happens. So here is my question for my whole thesis and for my whole presentation. It is, can we reconstitute these rapid calcium powered contractions in our lab? And we are thinking about using the myelin as a start. Myelins is being found in different organisms and different cilia. And this mechanism has been studied not only the spiral stomach, but also the verticilla and also the tetrahemiter. But we have a problem here. It's like nobody has done the myelin sample size purification in enough quantity so that for us to do the experiment. And then our collaborator, Dr. Hans, who has studied the tetraheminar and the calcium responding tetraheminar protein for several years, and who gave us the solution. So we find out the TCB2 and other calcium binding protein are homologs in this system, and Dr. Jerry Hans has studied the tetraheminar and TCB2 protein for really long decade, and he know exactly how to synthesize and purify it, and so that we could do more experiment for this. Let me just introduce this is a method part. Is like Dr. Hans helped us and trained me how to do all the purification and the synthesize, and he's a master who is helping us and doing all this synthesize for the past years. And okay, right now let me play a video. The first time when I joined the lab in 2019, this is the video start recruiting me and showed me. So what happens here is what you're seeing here is Jerry inject the calcium from the left-hand side into the TCB2 solution, and the whole system is in the glass slide into sandwich structure. What you're seeing here is once the calcium get into the system, the TCB2 goes through this kind of phase transition from solution to the gel. And if we zoom in to see the boundary, what happens here is like we can say it generates a wave going back to the system. And this is like so exciting for me because this is exactly this example showing the TCB2 protein could generate enough contractile force and could help us to reconstitute this calcium-powered system in our lab. But I have tried this experiment in our lab for quite a long time. They have some problem. It's uncontrollable. It's hard to, because when we are pipette the calcium into the system, it's really hard to control the force and to repeat everything.
You have to admit it like someone else.
Can anyone else help me admit?
You can disable the waiting room.
It's just,
Okay.
it's not this one. Okay.
They're admitted it's waiting. There they go.
Thank you. So it's uncontrollable. Is there someone else?
Just click back again. Yeah.
So I want to control the TCB2 in a better way. And in my thesis, there are two small parts I want to cover. The first one, how do I control the TCB2? The TCB2 with the calcium, it will just self-assemble into the protein network. The left-hand side, the image you're seeing is a TEM, and here's my, okay, it's a TEM, and you're seeing the TCB2 without the calcium. You're seeing these 200 nanometer filaments forming here. And once we are adding the calcium into the system, the whole network formed. And basically what happens here is like a TCB2, calcium is as a linker to connect the whole TCB2 system from the filament to a micrometers network. And yes, so what is TCB2? I want to give like some simple introduction for the molecular structure here. So the TCB2 is a protein which is 25 kilodalton. So it's the same size as green fluorescence protein. It's pretty small. And you can see it's like at the C terminal, at C and the N terminal, they have 4 EF hands and forming the two pockets and the C and N terminals here. And all this image I'm showing here is by AlphaFold and by Dr. Jerry Hans. And you're seeing here is like once the calcium binding to the system, you can see this conformational change. It's they're driving the N terminal to the C terminal. So this is the only molecular structure we know about the whole system yet. And we don't have the pro-EM or better or more highly resolution for the system. So our story is based on this kind of observation from here. To control it, what we do is we talk this problem with Scott, and he give us a really nice suggestion. Is using a photoliable cage. So this photoliable cage, it means like they have a photoliable bond here. If you stay in here, what happens here is like once we are shining on the UV light to the system, this bond will break, and then they will release the calcium to the system. And for this specific chelator, it has been widely used in the like optogenetics and neuroscience. It is great fit for our project because it can raise the concentration from nanomolar to the micromolars immediately. I'd like to give an example how I do this. So this is a setup I built back in 2020. So what we do is we mounted a device. It's called digital macro mirror. You can think about it as just a projector you mount into the microscope. And what we do is like we also including one dacron mirror into the system. So our lab, our microscope in the upstairs labs, they can do is like they can not only do the light activation from the projector, they can also doing the confocal and also the fluorescence image at the same time. So in this way, we could do a more quantified way of knowing the system. And right now, let me show you interesting part. So I'm going to give a light pattern of a circle here, and what you're seeing here is the TCB2 system, how they respond to my light pattern showed from the digital mirror here. And you can say it's exactly the same shape of my light projection. And if we do a chemograph, what I mean is like from the middle line we're drawing, we can see around the 100 seconds of the light control, light activation, we can see the network start to growing from the middle light activation part to the outside. Okay, there is going to be more fun with all these projection patterns through the computer. The computer can transmit all this pattern I want to do. So this is the first one. I give it to you. It's a sharp 5-pointed star. And you can see the system will respond to the 5-pointed star immediately and react in the seconds. And more than that, I can also give like a sequence of the light pattern here. So here I give a clockwise trajectory here, and you can see the system dynamically responds to each pattern and forming this beautiful clock shape here. This is the setup I already introduced, so I want to do a quick summary for you to understand how this setup works, and it will help us to moving forward. So the first step, what we do is like we use the light to activate the chelator. The chelator will release the calcium. Second, the calcium will start to binding to the TCB2 or the protein we're starting with. And third step, once they're binding to the once the calcium start binding to the TCB2, what happens is like the TCB2 will use this cross-linker to form a really dense network here. I'd like to introduce one more type. So for the setup we're using excessive chelator in the system, so that when we switch off the light, we can recapture the calcium through the system. So that in this way, we're kind of make a closed loop for our system to reuse. And this is a full picture of the setup. Okay, right now we already know all the chemical components in the system. Let's just saying how them interact with each other. And to do that, let's start with something simple. It's still using the 75 micron, like a circle disc in the middle. And what we're seeing here is it forming a network, slowly diffuse, slowly growing. It is because the calcium diffusing outwards. So let me play the video again. So what happens is like in the light yellow pattern here, then I will release the calcium, and the calcium will diffuse from the yellow part outside, so that in this way, they will trigger the reaction happening and they will forming the dense network. And moreover, what we find out is something else. This is the same video, but I just treated it with a PLA. And what you're saying here is along the boundary, there is a lot of arrow happening. And what that arrow means, it means like every single filaments inside, they segmented and they track it. And what happens is, if we're seeing here, is along the boundary, what happens is like they forming the network and the network contract back. This is really interesting to me because there are two processes happening at the same time, but at the different direction. Chemically, what happens is calcium diffuse from inside to outside, deforming the network. Mechanically, what happens is once they're forming the network along the boundary, actually they mechanically contract the other direction from outside to inside. Let me a little bit zoom in to just look at this data more carefully, and let me explain how I get the data. So what we are using is a PLA, which is use a particle image tracking system. What happens is like they compare the image at T0 and with the delta T. What happens is like they segment each particles and they're comparing which is the direction happens here. If we're in our case, what happens here is like you will see the small filaments inside here, and each arrow they will comparing the T equals 0 and the delta T, and then they will giving us the signal. It's like okay, where the particle goes. What I'm showing you here is the example of the T equals to 30 seconds. And what we do is like instead of using x, y coordinate, we change to the radical coordinate. And we can calculate this to the radical velocity. And for the radical velocity, what we could do is like we can compress this one image figure to this heat map. So this heat map I'm showing you is a much dense information. It is like a 1600 seconds image. I condensed it into one chemograph here, and maybe I could help you to go through a little bit better. So right now, the image I'm showing you is 30 seconds. And if you see the gray slice I'm pointing out here, this is exactly the slice is corresponding to the image I showed here. And apologize for the confusing part. It's like in the chemograph, I use a different color, the dark red color. So for showing the contraction happening here. So the blue arrow you are showing here in the chemograph here actually is the dark red color. And what you are showing here, what you are seeing here is inside of the pattern, there is nothing comes out, and at the parameter, you can see the strong arrow happenings at the parameter. What you can say from this chemograph, what happens here is like if you can see my mouse, what happens here is like from 0 to 105 microns. If I go from 0 all the way, nothing happens, and around the 50 or the parameter happens here, you can see the dark red line. So this point actually is just what I do is like an average at the same radicals, the velocity, and you can see it has a really strong velocity happens here. Contraction wave happens along the boundary, and it happens from the 0 second to 100 second here. By the way, I just want to mention we also do the simulation for the system, and the simulation you can see from here, it matches the experiment data pretty well. Okay, for the whole system, I want to introduce one more layer. It is a mechanical layer. It is not only the chemical happening here. Right now, the mechanical also happens here. What is the molecular structure? Because I just talked about the molecular will just doing the conformation change and they will shrink after they binding to the calcium. So what we assume is what happens here is once they change their conformation, they will have a new rest length. Or if we're thinking it's a spring, what happens is before they have a rest length, and after they're bending, they have a new rest length. And to make it connected to our chemical modeling, what happens is we're assuming, of course, here is a different share rate, but if I want to give a simple example, what happens here is we assume the local spring Young's modulus is directly linked to the local concentration. It means if I have more TCB2 at one concentration, it will have a stiff Young's modulus. It will have a stiff contraction happening there. And I have another slide for the modeling part, later if I can just, if you have a question for that, I can just elaborate a little bit more for that part. So basically what we do is we have chemical model and we link it one more layer to the mechanical model. So what happens here is our collaborator, Dr. Carlos Floyd, get all the information from the experiment. And then what he do is he is trying to recapture all the behavior I see from experiment from the modeling part. And from the modeling, what we do is first, because we have the control of the light through the computer. And now what we can control is we can release the calcium through the control of the light intensity. And the next step is since we link the using the reaction diffusion rate to link the calcium concentration to the TCB2 concentration, so we can get the TCB2 concentration in real time. You might wondering, because you already have the green fluorescent type, why you don't just directly using the green fluorescence to quantify the TCB2 concentration. I have to say it's really hard and it cannot give me the concentration at each specific location in real time. And one more layer, what we do is since we have the local concentration of the TCB2 and we assume the Young's modulus is linearly linked to the calcium TCB2 concentration. What we could do is we could have the displacement of the string field immediately. And with the string field, when we do the derivative over the time, we can have the local velocity and the H positions immediately as well. In this way, we could know our system. What happens is both chemically I introduced and also mechanically I just mentioned about it in a really in a great way and in a controlled way. Okay, just now I just showing all the system, and I want to mention one more time is this system is not only can be controlled using the light. The fascinating or the great part of the system is we can reset the system. What I mean reset the system is because we introduce the excessive chelator into the system. What happens is when we switch off the light, the excessive chelator will reband to the calcium and they will remove the calcium through the system. What I mean is it allows us to do the rapid triggering with the pulse light. Just now, the example I showed you, I always give a continuous light pulse to the system. And right now, I'm going to show a little bit fancier. What I do is I give the pulse light to the system and saying not only one time actuation, but I want to saying, can we use the system in a more efficient way and in a better way? So the pattern I'm giving you here, if you can see my mouse here, instead of a continuous pattern, I'm going to give a pulse-like pattern here. And maybe I could ask you to imagine what happens to the system. Will they respond to the pulse light, or do they will just ignore it and start to growing whatever in their like way? Okay, so here So here is the example I gave in. The light pattern I gave is on the right-hand side here. You can say it's a pulse light. I may ask you to look at the video slowly and carefully when I play it. The yellow part is the light activation pattern I gave. And this is the device videos I recorded in time. And what you're seeing here is like every time when I activate the system, you can see this wave happens here. And why it happens here, or what happens here is like, let me just go use the camel graph again, trying to help you guys to understand. What I gave the light is like I give 1 second light on and 29 second off. If you're saying the camel graph, what happens here is like when I'm giving the 1 second light on, they will forming the network immediately. And then when I turn off the light, what you're seeing is it slowly disassemble. And the whole network kind of disassemble. But when I is going to recharge or we're going to give the signal again, you can say it's forming the network again and disassemble, forming the network again and disassemble. But in a general trend, you can say the network is start to growing through this slope. And why it happens like that, I want to mention like one of the bending rate here. Because when we are turn off the light, when we are turn on the light, the chelator doesn't work anymore. But when we are turn off the light without the light, the chelator's bending rate is much higher than our TCP2 bending rate. It means it's when I turn off the light, it's much stronger competitors in the system, and they are getting the calcium from the TCP2 into their system. And we do the test for their like repeatability. It's like after 200 cycles, they are still respond to my light, and they can just be used pretty strongly. And right now, I want to show you. Oh, I got stuck. The video is huge, so every time when I play it, like sometimes it just gets stuck here. Let me try again, seeing whether it's loading well. Okay, so basically in this PIV, just maybe imagine you already see the PIV. What happens here is like every time when I'm triggering a system, you can see they are tracking the errors happening all the time. And using the data I don't show it now, but what we can do is like we can do the camel graph for the pulse protocol again. And you are seeing what happens here. Every cycle when I'm triggering the system, we can see the dark red color happens here. It means like along the parameters, they have this strong contraction happens. And let me continue. Let me trying to just prepare, just to compare the pulse protocol and the continuous protocol together. What happens here? What you're seeing here is like here is a continuous protocol. What I do is like I give 100 second light on, and you're seeing, and then they don't showing any dark region anymore. It means the system slowly dying down. But if I give the pulse to the system, every time I pulse the system, it's kind of like I kind of recharging the system again and again. And it start to giving us like this response stronger and stronger. So what I mean stronger is if you're seeing the dark color here from the beginning to the end, there the color becomes darker. It means the contraction rate get higher and higher. To do one more like a quantification to understand that, what we do is like we define the velocity, the radical velocity. If it's above 0.2 micron second, we define it as active region. And for the active region, what we find out actually for the continuous protocol, they're only showing this active region in the initial 30 seconds, and then they slowly dying down. But for the pulse protocol, what we are showing here, every time we are pulsing the light, it giving the signal to the system, and they are just pumping up the whole system. And you can say, actually, they have the trend to increase the active region like slowly. So you might think it might be a little bit hard to compare, or how should I compare this? Let me give an example. This is all the experiment data. So on the left-hand side, what I gave it to you is a continuous protocol at 30 seconds. So what I mean is like I gave a 30-second continuous light. And on the right-hand side, what I gave you is like the 30th pulse, which means what happens here is like I gave 1 second light on and I waited, and I gave another 1 second light on. So what I comparing here is like I gave the same time of the light on time or the activation time. And you can see compared to the right and left, you can say for the pulse signal, it's much like thicker parameters happen there, and also they are much stronger happening here. So how we understand that what happens here is like you can think about you have a spring. When we are kind of stretching it and we let it down. With a continuous light, it means you're just stretching it all the time. And with a pulse light, you're kind of like stretching, relaxing, stretching, relaxing. And what happens is like along the boundary or along the parameter, what happens is like you will keep them under the active tension all the time. And this way we could generate a much larger and faster active boundary. So not only the active boundary, we also find actually with kind of this pulse light, we find out something more interesting. What happens here is if we continuously observing, oh, the PIV works, great. So if you look close to the PIV, what happens here is you can see there is like two parts. If you can see my mouse here, what happens is like the inside is trying to going outward, and the outside is trying to contract inward. This is really interesting to me because what I'm thinking is like as for you thinking a contraction or contracting system, you are always thinking is maybe just contracting and towards to one direction. But how could this have also the expansion at the same time? And then we look really close to the system, and what we find out, actually, it also linked to the local concentration. This is the experiment what I did with 1 micron polystyrene beads. I embedded the really small micron beads into the system, trying to visualize it. And what you're seeing here, if I play the video, active from the middle, and if you are seeing along the boundary, actually they have a huge accumulation, and then they have a dark region here. What means is like if we look at the concentration here, it happens is like first they accumulate along the boundary, and then they have a drop here. And in this way, this kind of accumulation, what happens is like because what we assume or we understand the system like the local contraction is directly linked to the local concentration. If we have accumulate along the boundary, it means they are more stiff in the middle, or they are more stiff along the middle line here. And for the inside, they are trying to contract towards to the much more stiff part, and also the outside, they are trying to contract towards to the center of the mass as well. That's why they give us this like the two direction opposite to each other. And we find out like this push and pull motion are really interesting, and it's not more than interesting. We can use it for something. Since we have the two direction control, what we do is we're thinking, okay, if you have the direction control, can we use it for something? So right now I'm showing you one video with the polystyrene beads embedded into the system. And what I do is like I gave the signaling from the middle. I gave the light contraction happens. And what you can say is the particle one start moving. And if I give another pattern, the particle one actually change the directions. And then I can also gave the triggering the different signal and triggering different particles at the different time as well. Please allow me to play this video one more time because I think this is one great example to showing you how this system can do. So let me play it one more time. Okay, here we go. So first, I'm triggering from the center, and you can say the particle 1 start moving. And it's also like you may wonder why it start moving at the boundary, because I showed in earlier slides, along the boundary, the contraction happens. And secondly, also you can see from here, it's like it's reversible, the whole system disassemble. And then I gave the signal to the right-hand side, and you can see the particle 1 start to change the direction towards to the center of the second projection I gave. And later, if I gave I also can do this kind of multi-illumination for the system. And using this system, what we could do is like we can program the trajectory for the different cell or for the different particle to do the different things. And in our paper, actually, Dr. Carlos Floyd also developed a reinforcement learning model for us so that we can design a closed loop to connect to our computer to the microscope so that we can do the in real time. So we plan to do the in real time analysis with the deep learning modeling so we can control this in a much better way. Okay, so right now, let me share one of my innovation or my dream experiments. I spent a really long time for this the cell droplets. So because just now what I'm saying is like, can we use this calcium driven network to do something to mimic the cell or to do something trying to kind of recapture this amazing contraction happens. So what I do is like I'm encapsulate the TCP2 into the droplets, and I want to see what happens inside of the droplets. Could they function the same as in the spirostom, in the tetraheminar, or in anything? But I have to admit, this is really hard experiment, and I have tried really long time. And but till today, I still could only summarize what I have observed and I have tested. So for the lipid or the droplet making things, what we do is there are different kind of liposome people are using to mimic the cell. One called is liposome. It has a double layer of lipid. And what happens here is like it's water in the water. And another way is much simpler. You can say it is just one single layer of the phospholipid, and it's not dual layer phospholipid. And what happens is it's water in all your droplets. And we have tried both ways, and we find out actually for the single layer water in all your droplets is a much the simplest way. And I'd like to show you some examples what I do here. So let me play the video here. What you're saying is water in all your droplets. And I'm triggering the signal from the bottom light here. And what you're saying here is actually first, I encapsulate the TCP2 and also the chelator successfully into the droplet. And you can say it start to gelation and react to the system immediately. But you might also notice there is a bright ring happens here, and you may wonder what is that if I just let my mouse happens here again. So actually what happens here, it is because it attached to the glass slide. We use the BSA to just pre-coating our glass slide, and it is really hydrophilic. And what happens here is if you allow me to just show you here, what happens here is actually when you are using the BSA to coating the slides, it is what happens here is like they are not fully forming a spherical, they are attached to the slides. And once we are activate using the light, because the light gives the energy to the system and they slowly spread out and evaporate to a small circle here. And then you might say, okay, why you don't use a much stronger, say like a saline or any other things to just kind of pre-coding the system. Right now, where I'm showing a really hydrophobic saline system, and you're saying, yes, you do not attach to the glass side anymore. But what happens is that it starts rotating and it gives us the 3D motion. And unfortunately, our lab, for me, it's like what I do is like I just do the projection and DIC recording. It is really hard for me to systematically study all the thing. So I already just summarize all my finding in my thesis, and I think there is like going to be more like a systematic study here. And I'm going to give one more example. So what happens here is you are saying a compartment forming in the droplet, and then if I triggering it, the system, you can say this kind of phase transition, this kind of solution to gel transition, actually they can kind of like reshape, change the shape of the droplet here. And if I play in here, what you're saying is actually they rotate and they start forming a small bubble at the circle here. But as you can see, it is really complicated. And I tried quite a long time, but I think maybe another PhD student will start to working on this in the future. So I have finished my first part and the second part, and this is the last maybe 10 slides of my presentation. And I want to end it with all the characterization I do for the system. So in the earlier slide, I showing, okay, this is really strong, but do we understand any mechanical property of the system? So I want to understand how strong is like deforming the network and how could we characterize it. So what I do is I use the optical tweezer to characterize the whole system. And how I do this is like I use a microfluid channel. And using this microfluid channel, what we can do is like we can freshly form the TCB2 at the right calcium concentration in the yellow box here. And then what we do is like we can embed it, the optical tweezer beads into the system. And when we are controlling the beads, what we can get is two information. First is of course the time, how much we move, but the most important is like we can tracking the bees using the tracking the bees velocity, and we can knowing the bees displacement. More important, when we are moving it, actually we can knowing the force we used to move the bees. And let me showing you one of the calcium concentration or like confocal image I did before. So what happens here is we inject or we having the different local concentration of the calcium from really low to really high. And what you can see here is like at 1 millimolar concentration, what happens is forming this kind of local clusters. And when we are increasing the concentration, we can see it start to forming this fully connect network. And if we're injecting too much calcium concentration into the system, what happens is like it start to having this rupture happens at the right corner here. And then I'm thinking, okay, since the calcium changed the protein network structure, so the mesostructure, it will definitely influence. It definitely will influence the property of the mechanical property of these different materials. So what I do is like I testing the the stiffness of the system with a different calcium concentration. And what you are seeing here is like with really low the local cluster concentration, actually we don't see any like stiffness of the system once we embed the beads into the system. And now when we are increasing the concentration and to the fully connect network, for example, the yellow one or the rupture network, we find out the stiffness of the network are much, much stronger compared to the local cluster or anything like here. And I do the experiment for like a lot of repeats to find out it's quite constant to see the trend. And another experiment, what I did is like, okay, this is a viscoelasticity model. It's not only elasticity. So what I do is I embedded a bead into the system, as you can see here. and then I move the beads from one direction to another direction. So let me play one video for you. I move it to 10 micron, and then I release it. If you're thinking about it's in water, once you move the beads, they will stay there. And if you're thinking about you embed the beads into a rubber, once you move the beads, it will bounce back. So what happens to our system? Let me play the second one. So with the middle, like a connect network, you're saying this is network, and that when we are releasing it, actually it's just bouncing back. And let me play the last video, which is like a really dense. So what you're seeing here, the network is the beast is kind of like jumping back, not fully original place, but they jumping back really fast and quick. And then it's like we're controlling using the calcium from the viscosity dominant region to the elasticity dominant region. Okay, right now is the last experiment slides I'm going to show you. So I'm going to give you a small quiz here. Right now you are seeing an experiment. I'm moving the beads from the, like I'm giving the two directional movement. First, I move to the left, and then I move to the up. I will ask you, do you think first, there is like there are two ways to going back. First, maybe they remember the trajectory they coming along, so they will go exactly the same trajectory back. The second way, maybe they don't go back anymore, or do you think there is another way? Will they just create a new way to bounce back? Maybe I will give you like a 4 seconds to think about that, and then I'm going to play the video. When I do the experiment, I thought like, okay, maybe they will follow the trajectory back. Let me play the video right now. And what you're seeing here, it's a trajectory. They are bouncing back towards to the diagonal way. And now when I check the literature, actually what they are saying is like the system basically memorized the elastic modulus along the both way, and then they will directly bounce back to the original place. And another thing I want to emphasize here is this recoiling velocity actually is huge. It's around 15, almost to 20 micron per second. And it shows the property of the system to behave kind of like storing the energy and performing this kind of like a fast movement as well. And yes, basically that's all my presentation. Let me do the conclusion slides for you one by one. First, we know the ultra-fast motion are driven by the active protein network. Second, what we do is like for this active protein network, we synthesize the cytoskeleton protein TCP2 from tetrahedral. And the third one is I built the optical platform to control this TCP2 network in our lab. And the fourth one is actually they show a lot of really complex behavior, but with just the reaction diffusion and linked to the viscoelasticity model, they can recapture all this complex behavior already. And it gives us a great model to continue our study. And the fifth one is actually we can already use these particles. We can use this network to transport the particles in the programmed way, and also they show the potential to be used in droplet to generate the force. And the last one is like the calcium concentration will change the network's structure and viscoelasticity. And thank you for all. And please just bear me for 1 second for my acknowledgement. And let me share screen again.